All right, in part two of this video, we're going to look at speciation and the methods that speciation occur. All right, so what is speciation? Well, the definition is pretty simple. It's the development of new species. It usually starts when a group from the same species is separated, all right, from the group. So we have some event separates a species into two different groups. Here's the reason that leads to speciation. It's really simple. A separated group cannot interbreed, and the lack of interbreeding eventually leads to two new species, to some kind of divergence. Well, why is that? Well, pretty simple, right? They're no longer sharing those mutations that are present in the population. So if we think about it like, here's our original group, and it's divided into two smaller groups that are separated. Mutations in group one are no longer shared in group two. So the change in time of this group is going to lead maybe to a bigger circle, right? And then this group could eventually lead to, say, a diamond, right? We'll just look at it like shapes. So when you can't share your mutations, when there's no interbreeding in a population, then they can change over time in ways that are no longer related. Now, how do we separate them? Well, there's a lot of different ways you can isolate a population. The first one is behavioral isolation. And this is when a population is separated by what they do. Geographic isolation is when a population is separated by where they live, which could be caused by a new obstacle if it's a small organism, or it could be caused by some kind of migration if there's a large organism. There's temporal isolation. Temporal comes from the word that means time, and that's when a population is separated by when they're active, so like throughout the day, or the time of year in which they breed. So let's look at each one of these. Behavioral isolation is pretty simple to identify. Different songs in different birds are used to attract mates. And when you look at the eastern versus the western meadowlark, they have a different song. So even though they look very, very similar, and they are both meadowlarks, the song that they use to attract a mate is different. So a western meadowlark will not respond to the song of an eastern meadowlark. So the mutations and alleles that are present in the eastern meadowlarks are not shared in the western meadowlark population, which can lead to, or which does lead to, independent change in each group. And you can kind of see that in the feather structure, especially in the yellow around the eye here. This has much more yellow than that bird there. So there's starting to be mutations and changes that are not present in both birds. Geographic isolation is typically from some kind of physical barrier. So a great example of that is the Isthmus of Panama. Couldn't get that one out. Before Panama was separated, right? So there was a path where you could go from the Caribbean Sea out into the Pacific Ocean. But since Panama is now a land bridge between North and South America, this is no longer allowed. So if we look at porkfish, which are a pretty common species in both the Caribbean and the Pacific, and we look at the porkfish that live in the Caribbean and we compare those to the porkfish that live in the Pacific, we can see that there's definitely a color variation there. And this has probably arisen from the fact that the water of the Pacific and the water of the Caribbean are very different colors. So if you're living in this Caribbean Sea, right up top, you have this nice blue color. It's pretty clear water. It makes sense that you in order to camouflage, you probably have some of these blue streaks. On the other hand, if you're living in greener water, those colors are more muted in order for you to be adapted to survive. And so the porkfish that lives in the Pacific Ocean has changed. Now, the fact that they cannot interbreed means that this coloration is more likely to continue, this blue coloration rather, is more likely to continue over here in the Caribbean porkfish, and it's very unlikely to continue in the in the Pacific Ocean porkfish. So that would be an example of a geographic isolation. Temporal isolation is best looked at with the spotted skunk. So we have the eastern spotted skunk and we have the western spotted skunk. The western spotted skunk breeds in the fall. The eastern spotted skunk breeds in late winter. So since they're not actively breeding at the same time, then they're not going to exchange their genes. And so this western spotted skunk, which has this much more defined whiteness in the underside of his tail, it's got more stripe-like body coloration. The alleles for those visual characteristics are not being passed along in the eastern spotted skunk, which is definitely more spotted, and the tail is clearly a little bit different. So since they don't breed at the same time, those differences are likely to increase. They are isolated by the time that they breed. Now, there's 4 kinds of speciation that can occur based on these isolation events. There's allopatric speciation, which occurs when there's a geographical barrier. There's parapatric speciation, which comes from spatial isolation that is partial. So you, we're not going to look at that one closely, but basically there's some kind of isolating event that separates the spaces where organisms are active. There's sympatric speciation, where there is some kind of genetic change, a polymorphism, so many mutations or many changes there that changes the genes of a species. And there's peripatric speciation, which is where the periphery of a species, the edge of a species, undergoes some isolation and changes. Now, all of these can be kind of visualized by these green and yellow circles. So the green circle is the original population. The yellow change is the new population, the new species that comes from the original population. So in allopatric, we have some kind of barrier that's formed. Different mutations in both sides lead to the right side here having mutations, the left side doesn't. And so this, change in the species starts to occur, and that's our allopatric speciation. In peripatric, we get some kind of new niche. That isolated niche separates the second population from the first. Mutations in the second one are not shared with the first population, and that leads to a different species. Okay. Parapatric, you have a new niche again, but it's adjacent. It's still with the original population. However, that adjacent niche requires different adaptations to be successful, perhaps, or a different breeding method that is required to be successful, or perhaps a different way to attract a mate. And so that group starts to isolate itself and eventually becomes a new species. You also have sympatric, which is, in the population, we have just this change in the genes to this group. They then are no longer able to reproduce with the original population, and this leads to speciation. Now, we study population genetics, or that genetic polymorphism, that last one. We study that sometimes, or there are people who study that, and we call it population genetics. And this kind of gives us an interesting look at evolution from a different definition, where they define evolution as the change in the frequency of alleles in a population. So as the relative amounts of alleles change, then the population genetics as a whole are changing. So we say that individuals don't evolve on their own. The population changes over time as the allele frequency changes. Now, we can measure that mathematically, right, with the Hardy-Weinberg equation. And the Hardy-Weinberg equation allows us to determine the frequency of alleles in a population. And when the equation remains at equilibrium, there's no evolution. If there's no mutation, no changes in the environment, all that good stuff, right? That would give us an equilibrium answer there. So here's the Hardy-Weinberg equation. It's not overly complicated. It compares the amount of homozygous dominant genes to heterozygous genes to homozygous recessive genes, and it sets that equal to 1. You don't need to memorize or solve the Hardy-Weinberg equation in my class. I want you to be aware that it's out there. I'm going to show you a problem with it, all right, where you take this pea plant population and, we look at their genotypes, we get their frequencies, all right. We look at their phenotype frequencies, and we look to find a total allele frequency there. And we can go in and we can try to solve that to see how close we are to either change or no change or whatever. We're not going to get into solving it. I just want you to be aware that the Hardy-Weinberg equation is how we measure the change in allele frequency over time. And this is what it looks like right there. All right. So hopefully, at the end of these two videos, you can answer the following questions. You know the difference in gradualism and punctuated equilibrium. You can give me some similarities about them, give me some differences about them. Make sure you know adaptive and radiation and convergent evolution. Okay, be able to give me some similarities and some differences. Think about coevolution, right? The bees and the colors on the flowers. Try to come up with some other examples that might be coevolution examples. I'll give you one to go look up, right, to start you out on the path. Look at the acacia tree and the ants that live in its hollow thorns, right? Look at that relationship. And the last thing is, from this video, hopefully you can explain speciation and the different ways that it can happen. Make sure you know the isolating mechanisms and the 4 different kinds of speciation that occurs, right? The allopatric, the genetic polymorphism, all that good stuff, okay? If you have any questions, let me know, and I'll be glad to help you out.